Okay, so this is a video on DNA replication, and DNA looks something like this. So this here is a double helix, and a double helix is a double-stranded molecule. And if you untwist a double helix, it's going to look something like this. So to get from the double helix to the ladder, you had to have a gyrase enzyme that would unwind the chromatin from histone proteins. Also then, the helicase enzyme would untwist the DNA into a flat ladder. DNA is a polymer comprised of chains of molecules called nucleotides. A nucleotide is made out of a single deoxyribose sugar, a phosphate group, and also one nitrogen base. The sugar-phosphate backbone forms the structural framework of nucleic acids, and this is comprised of, as said in the title, sugar, so the deoxyribose sugar, and the phosphate group. They are held together with covalent bonds. The nitrogen bases hold the two DNA strands together with hydrogen bonds. As you can see, there are two hydrogen bonds between adenine and thymine. There are three hydrogen bonds between guanine and cytosine. You will notice that the two DNA strands run anti-parallel to each other. This means that while they are parallel, they run in the opposite directions. The DNA ladder is unzipped along the nitrogen bases. This is done by DNA polymerase III, which is an enzyme that comes along and breaks the hydrogen bonds between the complementary bases. The antiparallel nature of DNA creates a situation where one strand is going from the 5' direction to the 3' direction, while the other strand is going from the 3' to the 5'. One of the main ways DNA's antiparallel structure affects replication is in the way DNA polymerases build the new strands of DNA. DNA polymerase is the enzyme that connects nucleotides to make the new DNA in this process. DNA polymerases only work in the 5' to the 3' direction, so in one of the DNA strands this is easy as it opens up in that direction. On the other side, however, the lagging strand, the enzyme must work in the opposite direction, meaning it can only build discontinuous fragments as the double helix unwinds. Single-stranded binding proteins, or SSBs, attach to the outside of the replication fork, which is just the active area of replication, to prevent it from closing. The parent strand, which creates a daughter strand that's going from the 5' to the 3' direction, is the leading strand. This means that DNA replication occurs here continuously. DNA polymerase III is the one that's creating the replication of the DNA. On the lagging strand, however, a different process is required to replicate DNA. The primase enzyme attaches RNA primer to at least two spots on the lagging strand. This enables DNA polymerase III to attach nucleotides between RNA primers on the daughter cell from the 3' to the 5' direction, discontinuously in small fragments called the Okazaki fragments. DNA polymerase I detaches the RNA primers once the DNA replications between them have taken place, and also replaces them with DNA nucleotides. DNA ligase then comes along and glues the DNA pieces together. So that's basically the entire process of DNA replication. There's a picture on the side that I'm attaching that's kind of a quick summary of what I just explained, so that helps.